The purpose of DNA replication is to ensure that each cell in an organism has a complete and correct copy of the organism's genetic data. Semi-conservative replication is a type of replication where each of the two new DNA molecules is composed of one strand from the original molecule and one newly synthesized strand. Replication of DNA is bidirectional, meaning that it starts from the origin of replication at the ta-ta box, where the DNA replication begins, and moves towards the fork in both directions on the DNA strand. Prokaryotes only have one origin of replication, while eukaryotes have many on their DNA. Helicase is the enzyme that separates the DNA molecule into two separate complementary strands, breaking the hydrogen bonds between the nitrogenous bases. The single-strand bonding protein keeps the strands from bonding back together, while topoisomerase relieves tension in the chromatin. Primase lays down a temporary primer so nucleotides can bind to a free 3' carbon. On the leading strand, DNA polymerase III then begins to add new nucleotides to the strand to create a continuous line of replication with no interruptions. DNA polymerase I then removes the temporary primer and replaces it with the correct nucleotides. Ligase connects the gap between where the primer used to be and the newly replicated DNA strand starts to form a phosphodiester bond between the two. On the lagging strand, multiple primers are laid down, creating Okazaki fragments where DNA polymerase lays down new nucleotides between primers. This is discontinuous replication. The monomer of DNA is nucleic acid. On the sugar of the monomer, there are 5 carbons. The first carbon is located at the point where the nitrogenous base and sugars are connected. The rest of the carbons move around the sugar clockwise, with the last carbon ending here. The phosphate group of the monomer bonds with the 3' carbon on the other monomers. DNA is a double helix. The nitrogenous bases face toward each other, as you can see. The sugar phosphate backbone is on the outside. In this model, it is yellow. The two strands run antiparallel to each other, meaning a 5' to 3' strand will be complementary to a 3' to 5' strand, and vice versa. According to Shargraph's rules, adenine will bind to thymine, and guanine will always
Deoxyribose has an
bond to
oxygen
cytosine.
only on the 3rd carbon, while ribose has an oxygen on the 2nd and 3rd carbon, causing it to be unstable, unlike deoxyribose, which is stable.
DNA is double-stranded while RNA is single-stranded, and in DNA they use the bases A, T, G, and C, while in RNA they use the bases A, U, G, and C. The two purines are adenine and guanine, which are double-ringed. The two pyridamines are thymine and cytosine.